Um, Good morning, ladies and gentlemen. First, I would like to thank the organizers for inviting me here and giving me this opportunity to present some of our research work in plant genome editing. So, as you know, genome editing technologies were invented some years ago. Since then, the technologies have been widely used to modify genomes of viruses, bacteria, plant, and animals. So these technologies actually also allow scientists to make very precise DNA changes in plant cells, and they could also allow us to improve crop traits for some good quality or high quality, disease resistant, and some stress tolerance. There are three major sequence-specific nucleases, in contrast to zinc finger nucleus and the tau effector nucleus, which requires substantial protein engineering work for each DNA target. The CRISPR-Cas9 system requires only a single change in guide RNA. So in this way, this system is simple, inexpensive, and versatile for genome engineering. And you know, all these sequence-specific nucleases, actually, they can make double-strand break at specific size, and most importantly, they can be programmable. They can be programmed to any particular DNA sequences. So these activities can be harnessed as genome engineering to allow cells to make specific or precise DNA changes. The double-strand breaks created by Talon or CRISPR, they will be, or they are, repaired by one of the two mechanisms, non-homologous adjoining and homologous recombination. So the cells actually they have ability to detect the breaks and repair them simply by pasting together the broken end of DNA with very tiny sequence changes. Or they can also fix the breaks by incorporating a piece of DNA or genetic material. So if we have the way to introduce double-strand breaks, then we could trigger cells to, with some disruption of gene or to incorporation of a new genetic material. So precise genome modifications are achieved by harnessing DNA double-strand break repair pathway. I'm going to talk about agriculture and genome editing technology. So we know crop improvement requires the constant creation and the use of this new allelic variants. Conventional breeding is limited by providing some new genes or new alleles to meet agricultural changes, whereas this mutation breeding requires time-consuming and expensive screens of a large population. So with respect to transgenesis, one of the limitations is the complexity of this regulatory process, because we know transgenic plants normally carry foreign genes which are inserted randomly into plant genome. So only a very big biotech company can afford to developing those profitable traits only in those very major crops. So then for genome editing as a new plant breeding technology or technique, they can actually accelerate plant breeding by allowing the introduction of precise and predictable variations directly in an elite background. So then I'm going to talk about, give some examples of using this genome editing technology to develop some valuable traits. Wheat is one of the most important crops worldwide. However, the genome of wheat is huge. If you look at the picture, you can see wheat has 3 subgenomes. Compared to the other stable crops, wheat genome is really big. It's 17.1 gigabases. So pod mildew is one of the most destructive diseases in wheat. So the yield losses can reach as much as 40%. So currently, this disease is controlled by using heavy fungicides. Pod mildew affects not only barley, not only wheat, but a variety of species such as barley, Rhodopsis, and tomato. So loss of function of MLO conferred resistance to pod mildew has not been broken since its integration into the other barley elite varieties, like 30 years ago. So what is MLO? MLO proteins are the negative regulator of plant defense to pod mildew disease. So in wheat harbors 3 copies of MLO. So this genetic redundancy made it very difficult to obtain those MLO mutants directly from conventional breeding or from selection techniques. So we hypothesize targeted editing MLO genes in wheat may provide the opportunity to breed varieties with broad spectrum and durable resistance to pod mildew. So in order to modify all 3 copies simultaneously, we designed a pair of talent to target the conserved region of MLO. And the fork 1 here, you can see it functions as a pair of scissors to make a double-strand break. And the talent construct was delivered into immature embryo of wheat by particle bombardment method. So after self-pollinations, we obtained all the combinations of mutant plants. So this triple mutant with all 6 alleles mutated was obtained, was recovered by twice self-pollinations. So then when plants grow, we test for their resistance. So here you can see the number of microcolonies from this triple mutant was significantly decreased compared to wild type and the other mutants. And consistent with this result, no obvious fungi growing on the leaves of triple mutant. So where you have abundant level of fungi growing on wild type and the other mutants. So actually, then this last February, the USDA declared that this pod mildew-resistant wheat line will not or was not regulated. So next, I'm going to give an example using this technology to accelerate corn breeding. We know this heterosis has been successfully exploited on corn breeding. However, these male sterile lines are still in great need for this hybrid corn seed production. So using CRISPR technology, we knocked out maize thermosensitive male sterile gene. And if you look at the mutant showed male sterile at high temperature, but male fertile at low temperature. So this indicates that the targeted mutagenesis induced by CRISPR-Cas9 can really create some agriculturally important trait in corn. And then the last example I'm going to give is to using CRISPR technology to edit rice. We know fragrance is a very important quality trait in rice. So the presence of BADH2 results in the synthesis of 2AP, which is a major compound of fragrant compound. So when the gene was disrupted and targeted by CRISPR, the mutant 2AP content of mutants line, those 3 lines, showed was very similar to this our positive control, or even higher. So our positive control harbored a natural mutation of this gene. So imagine those fragrant mutant plants, they differ from their counterpart with very tiny changes in one gene in entire genome. So, but otherwise, it's same. So then we sequenced the DNA of those mutants, and we found that the DNA mutation occurred only in the place where the double-strand breaks were introduced. So this method provides rice breeders with some new method, new way to breed fragrant rice. So if you look at this conventional genome editing, we first delivered CRISPR or TALEN cassette into plant genome, which cut chromosome, introduce double-strand break, and lead to modifications. Meanwhile, this cassette will become integrated into plant genome. So after segregation away, like in T1 generation or T2 generation, then those nuclear transgenes will be segregated away, and no foreign DNA remains in mutant plant. But still, there are some disadvantages of this system. Firstly, this integrated CRISPR cassette or TALEN cassette will or may increase off-target effect. Secondly, for those vegetatively propagated plant species, such as potato, they don't have a chance to segregate away this transgene. And also, we often found at the double-strand break, there are some mutations, there are some small DNA insertions. So then we developed an efficient DNA-free genome editing method, so by which the mutant plants are generated from transiently expressing CRISPR-Cas9 protein. So we know under normal condition, protein will not become integrated into plant genome. So then we take example, we take like a GW2 gene, which has 3 copies in weight, and we designed a sgRNA, which match perfect with B1 and D1, but exists one mismatch. So if you look at the graph, you see all these 28 mutants induced by CRISPR-Cas9 protein, we couldn't detect any off-target mutations in a genome. And by a side-by-side experiment from the similar 28 mutants produced by this CRISPR-Cas9 DNA, 24 of them carried mutations in a genome, which are off-target. So then we know by optimizing genome editing technology, we have a chance to increase the biosafety of genome-modified plants. We start with stably expressing CRISPR DNA or transiently expressing CRISPR DNA or transiently expressing CRISPR RNA. To last, we use protein. So then the off-target effect will be decreased, and there will be no DNA recombination. To conclude my work, I would say genome editing can effectively induce targeted mutation in plant genomes at a very precise location, and the technology can be used to target many alleles simultaneously, and the system is very versatile, and the system is really good and cheap to use for small and underutilized crops. The transgene construct can be segregated by crossing, and for genome-edited final product, some of them, they are identical to the mutant obtained by conventional mutagenesis. To future perspective, we know CRISPR-Cas9 protein-mediated safe editing will become the major choice. And the system can really reduce costs for precise and efficient molecular breeding. And the product produced by RNP or produced by conventional genome editing, but without transgene, can really or greatly reduce regulatory requirements. And also by or through, like today, knowledge sharing, we could ease public concern about gene-edited crops. Thank you for your listening.